Before we hear about the actual process of translation, I wanted to introduce you to the cast of characters, so to speak, those molecules that are going to be required for the process to occur. Among these are the mRNA, and we've learned a lot about how the mRNA is produced. We've heard that the mRNA in a eukaryotic cell is going to be modified in the nucleus by the addition of a 5' cap, denoted by the red rectangle, at the 5' end, and a poly A tail, denoted by the black rectangle, at the 3' end of the mRNA. Once those modifications have been made, the mRNA can leave the nucleus for translation in the cytoplasm. A couple of other things about the mRNA that I wanted to point out is that there are these two regions that are labeled the 5' UTR at the 5' end and the 3' UTR at the 3' end of this mRNA transcript. These UTRs are untranslated regions of the mRNA that will not be quote-unquote read by the ribosome during the process of translation. Both of these regions are going to help in the process of translation by making it more efficient. And they're also going to be responsible for contributing to the longevity or lifespan of the mRNA. The 5' UTR is also the region of the mRNA that is going to be initially recognized by the ribosome prior to initiation of translation. In addition to the mRNA, I'll introduce you to the tRNA. The tRNA is denoted by this blue blobby region. I'll call it a fork because at one end of the tRNA, there is going to be an anticodon that will associate by hydrogen bonding with the mRNA. I say at one end, but in fact the anticodon is actually in the middle of the tRNA. At an end, however, in fact the 3' end of the tRNA, an amino acid denoted by this sort of green sphere will be added. Amino acids are added to tRNAs by a group of enzymes known as aminoacyl tRNA synthetases. In this case, the aqua-ish blob is meant to represent the aminoacyl tRNA synthetase enzyme. And because the amino acid that it will attach to the tRNA is tryptophan, this enzyme is specifically known as tryptophanyl tRNA synthetase. Tryptophanyl tRNA synthetase will recognize the anticodon of the tRNA, and it will know that this tRNA is meant to be charged with or linked to the amino acid tryptophan. It will catalyze the formation of a covalent chemical bond between the hydroxyl group associated with the carboxyl terminal end or side of the amino acid by linking it to the tRNA. In this way, when the tRNA that has been charged with an amino acid is brought to a ribosome, hydrogen bonding between the codon for tryptophan and the anticodon with this particular tRNA will bring, if you will, the appropriate amino acid to the ribosome. Looking at the chemistry of that covalent linkage, we see on the left-hand side that chemical bond between the carboxyl terminal end of the amino acid and the tRNA. On the right-hand side of this lower image, we see that the hydroxyl group is linked to the 3' end of the 3' terminal nucleotide of the tRNA. the dehydration synthesis reaction will allow for the OH from the 3' end of the nucleotide and the H, if you will, from the hydroxyl group on the carboxyl terminal end of the amino acid to produce water, just like every dehydration synthesis reaction we've seen thus far. Looking at the sequence of a tRNA, we see that indeed the anticodon, denoted in pink in the blue box at the lower part of this image, is in the middle of a tRNA. You'll also notice a few wonky looking letters in this tRNA. For example, right to the right of the anticodon, you'll see a little pitchfork looking thing, and that is a psi, I think, or C, Greek letter, and that is meant to denote that this tRNA nucleotide has been modified from one of the standard nucleotides that we know are in RNAs. This particular anticodon encodes for the amino acid phenylalanine. So the phenylalanyl tRNA synthetase will bring phenylalanine and charge this tRNA with that amino acid. That amino acid phenylalanine will be linked to the 3' adenosine of the tRNA. This particular tRNA terminates in the sequence CCA, and indeed all tRNAs terminate at their 3' end with the same 3 nucleotides, CCA. It is the 3' end of the adenosine nucleotide of all tRNAs to which the amino acids will be covalently linked by the enzymes that we looked at in the previous slide. This is a two-dimensional image of a tRNA, and this particular tRNA will be charged by alanine tRNA synthetase. Again, alanine will be linked by dehydration synthesis to the 3' end of the adenosine that is at the 3' end of the tRNA. Looking at the association between the anticodon of the tRNA and the codon on the mRNA, I want to highlight that the association, or the hydrogen bonding, or the base pairing, is going to be anti-parallel, just like the 2 strands of DNA are bonded in an anti-parallel nature. In other words, when we read the mRNA in the 5' to the 3' direction, it will associate with the tRNA in the opposite direction. So the GCA codon on the mRNA will bind to a UGC anticodon, reading both of those sequences in the 5' to 3' direction. The other thing that I want to highlight here is that the linkage at the 5' end of the anticodon, the 3' end of the codon, is not always as specific as it is shown here between the UA base pair. Indeed, that 5' in the anticodon, 3' in the codon position is known as the wobble position. In the previous slide, you saw an AU base pair, and in fact, whenever there is an A in the codon, there will be a U in the anticodon. However, if there is a U or a C in the codon, you'll note that there are more than one possibilities for the nucleotide that will be in the anticodon. Of course, the typical Watson-Crick base pairing that we would imagine is a possibility, but other possibilities exist. You may be surprised by the I as a possibility. I stands for inosine, and it is a modified nucleotide base. Finally, we've seen the mRNA and have been introduced to the tRNA and the tRNA's association with the amino acid. I now want to talk about ribosomes and the rRNAs in them. This image depicts a bacterial ribosome, and just like a eukaryotic ribosome, the prokaryotic ribosome is made up of two subunits, a small subunit and a large subunit. The numbers that are associated with these subunits are Svedberg units. Svedberg units denote the size of the subunits, and Svedberg is a guy who came up with these size units based upon how these ribosomal subunits and their component parts sedimented in a centrifuge tube. The small prokaryotic ribosomal subunit is 30 Svedberg units, or 30S, and within that 30S small prokaryotic subunit, there are a number of proteins as well as a single rRNA. The rRNA itself is also sized in Svedberg units, and this rRNA happens to be 16S in size. The 16S rRNA associates again with a number of proteins to make up the small ribosomal subunit. There are two rRNAs, a 23S and a 5S rRNA, that are combined with a number of proteins to make up the large prokaryotic ribosomal subunit that happens to be 50S. When those two smaller subunits assemble to create the intact prokaryotic ribosome, 50S plus 30S creates a, oddly, 70S ribosome. The construction of a eukaryotic ribosome is similar to that of a prokaryotic ribosome. There are going to be two subunits, a large and a small, and just like in the prokaryotic ribosome, a single RNA, in this case an 18S rRNA, will combine with a number of proteins to create the small ribosomal subunit. The small ribosomal subunit in a eukaryote is 40S in size. 3 rRNAs combine with a number of proteins to create the large ribosomal subunit in a eukaryote, and that large ribosomal subunit is 60S. So both the small and the large ribosomal subunits in a eukaryote are larger than they are in the prokaryote. That creates an intact ribosome that is indeed larger than the prokaryotic intact ribosome. 60S plus 40S in a eukaryote creates an 80S intact ribosome. Regardless of whether we're talking about a prokaryotic or a eukaryotic ribosome, when it is fully assembled, there are at least 2 binding sites for tRNAs. Those are the P site and the A site. The P site stands for the peptidyl site, and the A site stands for the amino acyl site. I say that there are at least 2 because some textbooks like yours includes a third site, the E site. Other textbooks completely leave out the E site because the E site is in fact the exit door. So while a tRNA will be moved into what your textbook calls the E site, it won't stay there very long because it will be kicked out of the ribosome in very short order. These are the players that are associated with translation. So after watching this video, we want you to watch a few more to show you how that process of translation occurs.